Hello class, this week we're going to be taking a look at the gross structure of neuroanatomy and an understanding of what makes up the brain. And before we get into talking about some of these topics, I believe it's important that we learn some vocabulary about the discipline so that we are all on the same page when we come to talk about what is going on here. We need to understand some basic principles about how the brain is organized, and then we can understand the rest of the course. At this moment, we will be talking really about the gross organization of the brain, and we're really talking about what can be seen at the level of the naked eye. That is, without the aid of the microscope. It is the overall big organization of the brain, and then we will look at specific functions a little bit later. But right now, we will look at some basic definitions. So when we speak of the central nervous system, we are talking about the brain and the spinal cord. And the nerves go along the spinal cord and into the skull through a hole called the foramen magnum. Now, it's very important that you recognize this, that the brain and the spinal cord must be continuous. So we cannot really have a break in the one. So the brain is housed in the skull and the spinal cord within the vertebrae. Now, we'll be focusing on the brain, but there is another aspect of the central nervous system, and that is called the peripheral nervous system. And this is any neural element that lies outside the brain and the spinal cord. So, for example, you have a long nerve that runs down the back of your leg called the sciatic nerve, and that is part of the peripheral nervous system. Any part of a nerve or nerve cell or process of a nerve that lies outside the brain and the spinal cord are part of the peripheral nervous system. And in order to refer to a number of different areas of the brain, we need to learn a number of different references. And these terms may not be familiar to you. So by way of example, the terms like rostral or caudal. So rostral, for example, means anterior, superior, or towards the face. So if I were to point to the frontal lobe, this would be rostral. Caudal then means toward the tail end, the back, the posterior. So if I were to point at the cerebellum, which would be a caudal area. Then there's dorsal, and dorsal means the upper parts or on top. So think in terms of the dorsal fin of a shark. Then there is ventral, and ventral means underneath or within. So now, when we refer to the spinal cord, the bony part, we can see what would be the dorsal, and the undersurface toward your organs would be the ventral. Medial then refers towards the midline of the body. Lateral refers to away from the midline of the body. So, I want to be clear that these terms are used in relative terms as well. So, if I were to put my finger in a spot on the brain, that spot is rostral to anything behind it, and caudal to everything in front of it. Now, remember, this is how neuroscientists talk to each other. So we need to be able to remember this language a little bit. Now, we're going to talk about the adult, fully developed brain when we discuss things. To be true, there are some significant changes that take place in the brain, and that the brain goes through human development. So just to keep things simple, we'll try to use the adult brain. Now, the adult brain can be grossly divided into 5 structures. So if we start from rostral to caudal, there are a number of major divisions that we can see. The first, most rostral, is the Telancephalon. And remember, cephalon just means head. So that should make it a little bit easier for you. Anytime you hear of something cephalic, that means it has to do with the head. So Telancephalon, then there is the Diencephalon, then the Mesencephalon, then the Mesencephalon, then the Myelin Cephalon. And these are terms you may have heard, you may not have heard about them. So when we talk about structures of the brain, we will always try to clarify where that structure resides. For example, a structure that lies within the Diencephalon. Where is that? Okay, so think in terms of the Telancephalon is what we see from the outside, this huge structure. And if we could look through this hemisphere, we would see the next section of the brain, which is the Diencephalon. And the Diencephalon is composed of two subunits, the thalamus and the hypothalamus. The next division of the brain we come to is the Mesencephalon, and this means midbrain. And it's approximately in the middle of the brain and lies just down a little bit. The next division of the brain is the Mesencephalon, and it consists of two structures, the cerebellum and a structure called the pons. The last structure of the brain is the Myelin Cephalon, and it is this structure that is connected to the spinal cord. So that's us talking about the different areas of the brain. So let's look at these divisions again, and this time I will use a little of the vocabulary to help us move along. The Telancephalon is the rostral-most subdivision. Essentially, cortex you see on the lateral and medial surface. The Telancephalon is the most human areas of the brain. It will be something that we will be very fascinated with. Just the outer layer of the Telancephalon is what is proper thought, what is properly thought of as the cortex. This is the seat of the mind. It is the outer part of the Telancephalon that makes up the cortex and is essentially what we think of as you or I. The cortex of other animals, it's much smaller than a human's. And the next area, remember we go from rostral to caudal, is the Diencephalon. So the portion that is caudal to the Telancephalon is the Diencephalon. It is subdivided into two different parts. The first is a dorsal division. Remember that means up. And this division is called the thalamus. The second is a ventral division. Remember, ventral means below, and that is the hypothalamus. So, what is the thalamus? The word literally means anteroom, the room before something. And that is actually what the thalamus is. All sensory information except smell goes through the thalamus, where it is then redirected to the areas of the brain, specifically the cortex, to give meaning to that sensory information. Almost all information that we get to the cortex must go through the thalamus. So we can see how that term anteroom, which thalamus means, is a very appropriate name because it is the place where information must go through. Now, the term hypothalamus, also part of the Diencephalon. Now, here I should make a side note that there are many different areas within the thalamus and also the hypothalamus. But we don't need to get into those nitty-gritty specifics right here in this course. There's lots of other time for us to take a look at the specific roles of those various areas in other courses. But in general, the role of the hypothalamus is to maintain homeostasis within the body. And it helps to get the body ready for demands from the external world and to ensure that the interior environment within the body stays within balance. Now, the next subdivision of the brain that we come to is Mesencephalon, or the midbrain. And it is in the middle of the brain. Actually, in a brain model, it goes almost straight across the top of the cerebellum. Now, this midbrain area has a lot of functions, but if we were to choose one thing that was largely covered by the mesencephalon, it would be reflexes. There are some very, very, very small structures within that midbrain. For example, the tip of my little finger would cover an area called the superior colliculus, also known as the rostral colliculus. Now, colliculus means little hill. So it is the rostrally located little hill. And it's involved in things like visual reflexes. So, for example, if there was suddenly a bright light over here, I might do a whole series of bodily movements, from lifting my hand to shielding my eye to turning my head to closing my eyes, and all of that reflex is governed by the superior colliculus. And this midbrain is guided by automatic scripts to respond to particular stimulus to help us protect ourselves. So it is a reflex that enables extremely fast responses. So we use our superior colliculus for this, but a toad would use his superior colliculus to localize prey in space, because it doesn't have time to process what a fast-moving fly is doing. So he uses his superior colliculus to do this. So when something dark and small and jerky comes into his field of view, his superior colliculus causes his tongue to stick out and to capture the fly. So the reflex function has a very different meaning from one creature to another, but the structure is similar. And the type of control is similar in the reflex. Now next we come to the Mesencephalon, which is made up of two main structures. The first is the cerebellum, which is literally the little brain. And this is a cauliflower-looking structure at the base of your brain, and it is responsible for the coordination of learned skill, of motor movements. Everything from tying your shoes to putting on your clothes to remembering how to sit down, how to stand up, and how to walk is controlled by this amazing structure. And the other part of the mesencephalon is the pons, a ball immediately in front of the cerebellum. And the pons, which means bridge, helps to process all of this. And so you can remember what the pons does in that it connects the cerebellum to the rest of the brain. It is the bridge to the rest of the brain. It acts as a bridge between the cerebellum and the brain. Now, I don't want to oversimplify things because these areas of the brain do many, many different things, but I hope you get a sense of the way that our brain works through this simplified explanation. Now comes the Myelin Cephalon, and that's the last structure of the brain. Is that place in the brain that becomes continuous with the spinal cord through that place in the skull, the foramen magnum. So the Myelin Cephalon also goes by the name of the medulla, or the medulla oblongata. And it's a basic area of the brain for survival, and is present in even the most primitive of organisms such as reptiles, and so oftentimes is called the reptilian brain. The term Myelin Cephalon literally means long white marrow structure. So basically, you do all of your thinking, planning, perceiving up in the rest of the brain, but in order to make a motor movement, you need to get the information down to your spinal cord. Because it is your spinal cord that is going to have the connections necessary to get the message out to your body. So all of that information has to travel through that area of the brain that is the medulla. Now, I want to add something more to that. Imagine you have a sensation in your leg, or your leg, you move your leg. Well, you know where your leg is at. In order for you to have a conscious perception of that, the information has to get to the brain, and this information must also go through the Myelin Cephalon. So, everything that the brain wants to communicate with the body has to go through the Myelin Cephalon. And everything that your body wants to communicate to your brain must go through the structures. So, when the early neuroanatomists opened it up, it was full of white matter, which is basically bundles of communication neurons. And that is why it is called the long white marrow structure. And that concludes our lecture in the gross anatomy of the brain. Now we're going to take some time and look at the overall division of the brain. We're not going to use those 5 cephalic divisions this time around. Instead, we're going to look at basic general ways of understanding the brain. So we're going to start off with the cortex. And we're going to be talking about the organization of the portion of the brain called the cortex. And that's the part that makes us who we are. That's part of that Telancephalon structure, for the most part. In order to really understand that, it's like trying to reverse engineer a simple circuit board. Only when you're trying to reverse a simple circuit board, you might be dealing with 20 different components, and that can be extremely difficult. So when you have a circuit board that has 100 billion components and several hundred various structures, that is so much more immensely complex. And that's what we're dealing with when we're trying to understand the brain, and particularly the cortex. So remember, the cortex in the brain has 100 billion neurons and hundreds of structures and dozens of different areas you need to understand in order to bring together a full understanding of how the human brain works. But what we do know is that the cortex really is the seat of everything we see, hear, feel, think, and what makes us us. And the elaboration of the cortex into its various functions is really one of the distinguishing characteristics of the human brain. It's because we have this elaboration of cortex that we can have a better understanding of the many various types of behavior that are specific to being human. For example, the ability to speak language is because of the organization of specific areas of our cortex that makes all of this possible. So what we're going to do is we're going to explore how we can make sense of the cortex and how scientists have used different classification schemes in order to partition up the cortex to make sense of it. So the cerebral cortex is the bark. Literally, cortex is bark. It's that outer part of the hemisphere. It's like tree bark. It's very variegated. And it is our outer sheet of cells, of gray matter. It's like a mantle of cells on the outside of the hemisphere. The cortex really appears to be wrinkled and folded because it gets compressed. It gets folded so that it will fit into your skull. If you were to look at the cortex of a rat, it is very smooth. By contrast, a human brain is all wrinkled. If you were to take the cortex and flatten it out on a table, you would see that it is about 2.5 square feet in size. So this is a look at cortical material. Now, the folds are called gyri. The singular for this term is gyrus. When you look at the valley in these folds, these are called sulci or sulcus and refers to the infolding of the cortex. If a sulcus is very, very deep, it is then called a fissure, a deep sulcus. If we are to look at the side of the brain at the midpoint, this is a sulcus, and its name is the central sulcus. And it is often referred in neuroscience as the central sulcus of Rolando, and the name is derived from the Italian neuroanatomist Luigi Rolando, who lived from 1773 to 1831, and obviously did a lot of work on neuroanatomy. It is just an infolding of the cortex, which is similar in almost all brains. So in all of us, our brains develop in a way that this major sulcus will appear. Now, our brains fold up in a sort of C shape to fit within our skulls. So we develop a deep fissure just below that sulcus, and it is named after Franciscus Sylvius, who lived from 1614 to 1672, and is known as the Sylvian fissure. Now, the cortex is very large in human beings, but it really isn't uniform, so the cortex varies in its thickness, and this variation is going to be between 1 and 4 millimeters thick. And I want you to just think about how thin that actually is. Extremely thin, and that's who you are. And it varies in what is called its cytoarchitecture. And the cytoarchitecture refers to how the cells are organized to communicate and to process information. Now, there are different cortical areas that have different layers of thickness. And in addition, neuroanatomists have identified 50 to 200 different types of organizational structures of neurons within the cortex. And that is what we mean by cytoarchitecture. Now, back in the turn of the 20th century, there was a researcher by the name of Corbin Brodmann. He lived between 1868 and 1918. And what Brodmann did was to divide the cortex into 50 areas based on how the cells were arranged in layers. So he found that there was a specific organizational structure of the cortex in the way that it's set up. We now know that the cortex in primates and humans can be divided up into approximately 200 different ways, just based upon cytoarchitecture. But it is Brodmann's system of cytoarchitecture that is retained in modern neuroscience. So sometimes you will see an area that is numbered within when you're reading an article, and that will be the Brodmann system. Now, the cortex can actually be divided up into a number of very simple areas, and we'll just take a look at those, because I will want you to know what these various areas are. There is the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. Now, the frontal lobe is an area of the brain of mammals located in the front of each cerebral hemisphere and positioned anterior to, that is in front of, or rostral of, the parietal lobe, and superior and anterior to the temporal lobes. It is separated by the parietal lobe by a space between tissues called the central sulcus, and from the temporal lobe by a deep fold called the lateral sylvian sulcus. The precentral gyrus, forming the posterior border of the frontal lobe, contains the primary motor cortex, which controls voluntary movements of specific body parts. The frontal lobe contains most of the dopamine-sensitive neurons of the cerebral cortex. The dopamine system is associated with reward, attention, short-term memory tasks, planning, and motivation. Dopamine tends to limit and select sensory information arriving from the thalamus to the forebrain. Now, a report from the National Institute of Mental Health says a gene variant that reduces dopamine activity in the prefrontal cortex is related to poor performance and inefficient functioning of that brain region during working memory tasks, and to slightly increased risk of schizophrenia. Now, as far as the anatomy of the central nervous system, on the lateral surface of the human brain, the central sulcus separates the frontal lobe from the parietal lobe, and the lateral sulcus separates the frontal lobe from the temporal lobe. Now, in humans, the frontal lobe reaches full maturity around 20 to 25. Marking the cognitive maturity that's normally associated with adulthood. A small amount of atrophy, however, is normal in the aging person's frontal lobe, and some studies in the early 2000s saw that there was some slight atrophy of people between the ages of 60 and 91. Now, there are a number of executive functions that take place in the frontal lobe, and they are involved primarily with the ability to recognize future consequences resulting from current actions. It helps us to choose between good and bad actions, or better and best. It overrides and suppresses socially unacceptable responses and determines similarities and differences between things or events. The frontal lobes also play an important part in retaining longer-term memories, which are not task-based. And these are often memories associated with emotions derived from input from the brain's limbic system. The frontal lobe modifies those emotions to generally fit socially accepted norms. And there's various ways that you can test the functioning of the frontal lobe. One of the most common ways is using a battery of psychometric tests, but simply looking at executive function. So the Tower of London executive function test, where you have to plan out a complex task, would be an excellent example of that. Then there is the parietal lobe, and the parietal lobe is the part of the brain positioned superior to the occipital lobe and behind, that is posterior to the frontal lobe. The parietal lobe really integrates sensory information from different modalities, determining spatial sense and things like navigation. Now, for example, it comprises the somatosensory cortex and the dorsal system of the visual system. And this enables regions of the parietal cortex to map objects perceived visually into body coordinating positions. The parietal lobe also plays roles in the integration of sensory information from various parts of the body. Knowledge of numbers and their relation to other objects. It also helps with coordination of visual spatial processing. So basically, it has to do with kinesthetic intelligence, the intelligence of body movement. Now, if there's damage to the right hemisphere of this lobe, in the loss of imagery, visualization, and spatial relationships, and neglect of left side space and left side of the body. So basically what will happen is you will lose control or awareness of part of your body even existing. Apraxia is a disorder of motor control that is often referred which can be referred neither to elemental motor deficits nor to general cognitive impairments. And the concept of apraxia really looks at what symptoms come from left brain damage, for example, that would influence right body functioning. So there's the contralateralism within the body. And that brings us to the end of this section. Hello class, and welcome to this lecture. My name is Dr. Neil Sogi, and for this lecture we're going to be taking a look at neurons and neurotransmitters. Now, a fundamental principle of neuroscience is that neurons are the structural and functional units of the nervous system. And this is known as the Neuronal Doctrine, a theory that was championed by the Nobel Prize-winning neuroanatomist Santiago Ramon Cajal. The doctrine established that neurons are the individual cellular units of the nervous system surrounded by a cellular membrane and separate from other structures like the cells in any other organ in the body. Unlike most cells in the body, however, neurons are specifically designed to receive and to transmit information. So, communication continually happens among 100 billion neurons in the adult brain. And despite some folksy notions that say we only use 10% of our brain, we actually are using all of it all of the time. It's just a matter of how hard we are using it and how much energy it is burning that varies. Now, a great advance forward in our understanding of the cellular communication in the nervous system came with the discovery of that axons generate electricity. And the electrical signal is generated at the axon helix and is propagated in an all or none fashion, meaning that there is no decrement of the signal as it travels down the axon. The signal jumps from node to node in between the myelin wrappings. The all or none electrical signal propagated along axons are known as the action potential. All action potentials generated by a single neuron are essentially the same in amplitude. The intensity of a stimulus is encoded by neurons by changing the frequency of the firing of the action potentials. Thus, an intense stimulus results in an increased firing rate of action potentials by a given neuron. The quality of the stimulus is the result of the stimulation of various types of neurons. Transmission along specific pathways in the brain and the interpretation by different areas of the brain occur, and this type of interpretation is especially true in the cerebral cortex. Now, to understand how neurons communicate, it is important to appreciate the internal and external environment of neurons and how changes in the distribution of ions, which are electrically charged atoms, can act as a signaling mechanism for transmitting information in the nervous system. Neurons in the body are surrounded by an extracellular fluid-filled space in which a number of ions are distributed. The inside of the neurons contains mitochondria, ions, and a variety of molecules. Ions, such as sodium, potassium, and chloride, are distributed unequally across the neuronal membrane, so that in this resting membrane potential, that is, when the neuron is not stimulated, the inside of the cell is more negatively charged than the outside of the cell. And this charge differentiation is maintained in part by specific molecules within the cell membrane. And it is extremely important that ionic concentration be strictly regulated by the nervous system. Abnormalities in these control mechanisms can actually lead to abnormal electrical discharge or to neuron death. Astrocytes, star-shaped cells, play a critical role in regulating the normal ionic extracellular environment in the CNS. And specifically, astrocytes, also known collectively as astroglia, are characteristic star-shaped glial cells, structural cells, in the brain and the spinal cord. They are the most abundant cell of the human brain, and they perform many functions, including biochemical support of endothelial cells that form the blood-brain barrier that protects the brain. They serve for provision of nutrients to the nervous tissue, maintenance of extracellular ion imbalance, and the role in the repair and scarring process of the brain and spinal cord following traumatic injuries. Now, when a neuron is stimulated, the distribution of ions, and thus charge, is altered across the membrane. If the inside of the cell becomes more positively charged, the cell is depolarized. And if movement of ions across the neuronal membrane causes the inside of the cell to become more negative, then the cell is said to be hyperpolarized. Action potentials are generated at the axon helix of the neuron and are propagated down the axon as rapid and transient charges in the membrane potential due to the movement of ions along at the breaks or nodes of the myelin sheath along the axon. This is referred to as saltatory conduction because the action potential jumps from node to node between the myelin sheaths. The velocity of this signal propagation is dependent on myelin, with larger myelinated axons conducting the fastest. Unmyelinated axons are the slowest conducting, a fact that helps us understand why a demyelinating disease such as multiple sclerosis can cause disability. Now, what happens when the action potential, that is the electrical charge, which is the same amplitude at every place, reaches the axon terminal endings? At the axonal ending, the action potential causes the presynaptic membrane to open channels, allowing an influx of calcium. And that sets off a cascade of events resulting in the fusion of synaptic vesicles with the presynaptic membrane and the release of chemicals called neurotransmitters. The neurotransmitter diffuses across the space between the presynaptic and postsynaptic membranes, known also as the synaptic cleft, to interact with receptors in the postsynaptic membrane. Now, what happens in the postsynaptic membrane as the result of the neurotransmitter binding? Well, neurotransmitter interaction with the postsynaptic receptors generates one of two outcomes. If open channels that are essentially proteins controlling the flow of ions across the neuronal membrane, or it causes binding with other molecules that ultimately produce charges in the membrane and the membrane potential in the dendrites or spines. The changes in the postsynaptic elements are referred to as synaptic potentials, and they are specifically generated in dendrites and in spines. They can also be depolarizing, that is excitatory, or hyperpolarizing, inhibitory, depending upon the type of postsynaptic receptor involved. It is the sum total of all synaptic inputs to a neuron which determines if it will fire an action potential. Unlike action potentials, synaptic potentials, are graded and decremental, meaning that their amplitude is not all or none. Their amplitude is dependent on the amount of neurotransmitter that is released. Moreover, as the signal is transferred down the neuronal membrane toward the axon helix, it degrades or diminishes. Thus, excitatory synapses on distal dendrites have less of an influence on firing the cell than those close to the axon helix. So, this is the basics of how neurons communicate in the nervous system. Now remember, it needs to be multiplied 100 billion times, then organized in specific ways, then placed in an environment and interact with it before you have a person and a personality. But you can see just how amazingly we are fearfully and wonderfully made with this process. Now I wish to transition a little bit and elaborate a little on the chemical part of this equation. One of the most important things I want you to think about as we talk about issues within this particular lecture is that particular neurotransmitters are going to be released by an individual neuron. That neurotransmitter will only bind to a certain postsynaptic receptor. So your neurotransmitter and your receptor have to be matched. The function of any area of the brain is dependent both on how it is connected via pathways to other areas of the brain and on the particular neurotransmitter that is used in individual synapses. There's a delicate balance and modulation of excitation and inhibition underlying the normal functioning of the nervous system. Many disorders can be understood in terms of a disruption of this finely orchestrated activity. Thus, damage to a single area of the brain disrupts function not only by removing that area from complicated circuits that underlie a particular function, but also by changing the relative influence of other excitatory and or inhibitory pathways. So what I want to talk about is some of the neurotransmitters used in the central nervous system and how synaptic transmission can be modulated. Now, neurotransmitters and neuromodulators play a critical role in the process of communication that occurs between neurons. They can be divided into a number of classes. And here we're going to just mention a few neurotransmitters and neuromodulators used in the nervous system that will also be important for understanding all manner of things like psychopathology and for addiction. Now, muscles are made up of proteins, and proteins are made up of amino acids. But there are lots of things that our amino acids are used for as well. 2 amino acid neurotransmitters are glutamate and gamma aminobutyric acid, also known as GABA. They are the major neurotransmitters of the central nervous system of all mammals. And most long axon neurons forming projection pathways in the brain use glutamate, which is an excitatory neurotransmitter. Gamma aminobutyric acid, GABA, is also used by long axon pathways. But it is also a major neurotransmitter used by interneurons. And it is an inhibitory neurotransmitter. Now, a little more about glutamate. Glutamate is the major excitatory neurotransmitter in the brain. The main motivation for the ongoing worldwide research on glutamate is due to the role of glutamate in the signal transduction of the nervous system of apparently all complex living organisms, including people. Glutamate is considered to be the major mediator of excitatory signals in the mammalian central nervous system and is involved in most aspects of normal brain function, including cognition, memory, and learning. Now here's the thing. Glutamate is toxic, not in spite of its importance, but because of it. Glutamate does not only mediate a lot of information, but also information which regulates brain development and information which determines cellular survival, differentiation, and elimination, as well as formation and elimination of nerve contacts, synapses. From this, it follows that glutamate has to be present in the right concentration, in the right places, for the right time. Both too much and too little glutamate is harmful. And this implies that glutamate is both essential and highly toxic at the same time. And it took a long time to realize that glutamate is a neurotransmitter. And it may sound astonishing, but it took the scientific community a long time to realize that glutamate is a neurotransmitter, although it was noted already 70 years ago that glutamate is abundant in the brain and that it plays a central role in brain metabolism. Ironically, the reason for the delay seems to have been its overwhelming importance. Glutamate is one of the extraordinary 20 amino acids which are used to make proteins and takes part in typical metabolic functions like energy production and ammonia detoxification, in addition to protein synthesis. And it was hard to believe that a compound with so many functions, and which is present virtually everywhere in high concentrations, could play an additional role as a neurotransmitter. Now, when it comes to neurotransmitters for understanding people, there are some very important what are called monoamines, biogenic amines, also called monoamines, in the central nervous system. And these include the catecholamines. And in the catecholamine category are some neurotransmitters such as dopamine and norepinephrine, and the indolamine, known as 5-HT, also known as serotonin. And these neurotransmitters are generally used by neurons localized to small nuclear groups distributed in the brain. Now, while the cell groups utilizing these neurotransmitters are small, their effect is large due to the widespread projections of these neurons. Now, a catecholamine is a organic compound that has a catechol, that is benzene with 2 hydroxyl side groups, and a side chain amine. And in the human body, the most abundant catecholamines are epinephrine, also known as adrenaline, and norepinephrine, known in the States as noradrenaline, and dopamine, all of which are produced by phenylalanine and tyrosine. And the release of the hormones epinephrine and norepinephrine from the adrenal medulla of the adrenal glands are part of that fight or flight response. And tyrosine is created by phenylalanine by hydroxylation by the enzyme phenylalanine hydroxylase. And tyrosine is also ingested directly from dietary protein. It is then sent to catecholamine secreting neurons, and here several reactions serially convert tyrosine to what is known as L-DOPA, and then to dopamine, and to norepinephrine, and eventually to epinephrine. And dopamine then is important for consciousness. For example, if you do not have enough, you go into a catatonic stupor. And there's a famous movie called Awakenings that deals with that. And it is also essential in rewards. So when you smoke, a key reward is the release of dopamine into the system. And 2 catecholamines, norepinephrine and dopamine act as neuromodulators in the central nervous system and as hormones in the blood circulation. The catecholamine norepinephrine is a neuromodulator of the peripheral sympathetic nervous system, but it is also present in the blood, mostly through spillover from the synapses of the synaptic pathways. And high catecholamine levels in the blood are associated with stress, which can be induced by psychological reactions or environmental stresses such as elevated sound noises, intense light, or low blood sugar levels. And extremely high levels of catecholamines, also known as catecholamine toxicity, can occur in the central nervous system. It can result in central nervous system trauma due to stimulation and or damage of nuclei in the brainstem. In particular, those nuclei affecting the sympathetic nervous system. So in emergency medicine, this occurrence is widely known as a catecholamine dump. Then there is the indolamine. We'll talk about the indolamine a little bit more. And that is serotonin, also known as 5-hydroxytryptamine, or 5-HT. And that is a monoamine neurotransmitter. Now, biochemically, this is derived from tryptophan, and serotonin is primarily found in the gastrointestinal, the GI tract. It's also found in platelets and in the central nervous system of animals, including humans, and is popularly thought to be a contributor to feelings of well-being and happiness. And essentially, serotonin helps to calm down the alarm system of the brain that are pre-reflexive in nature, meaning that they make up our interpretation of things. That is why one of the main ways of treating both anxiety and depression is to increase the amount of serotonin available in the brain. And this is done by inhibiting the amount of action that the reuptake functions of the brain for serotonin can do. Now, interestingly, being happy helps protect against things like heart attacks. And when there is more serotonin, and that's connected to platelets, the better the entire circulatory system works. And there's some interesting work by someone by the name of Charles Nemiroff that has looked at that more intently. Now, other things that are of importance are neurotransmitters like acetylcholine, which is used as a neurotransmitter by a few cell groups and some interneurons in the brain. And acetylcholine is an organic polyatomic ion that acts as a neurotransmitter in both the peripheral and the central nervous system in most organisms, including humans. And acetylcholine is one of many neurotransmitters in the autonomic nervous system, and the only neurotransmitter used in the motor division of the somatic nervous system. So sensory neurons include glutamate and various peptides at their synapses. Acetylcholine is also the principal neurotransmitter in all autonomic glia. Now, one well-supported function of acetylcholine in cortex is increased responsiveness to sensory stimuli, now a form of attention. Phasic increases in acetylcholine during visual, auditory, and sensory somatosensory stimulus presents what has been found to increase the firing rate of neurons in the corresponding primary sensory cortices. These are only the cursory introduction to neurotransmitters. There's over 60 different neurotransmitters, and each one has multiple and varied functions depending upon the location of the neurotransmitter, the level of the neurotransmitter, and the type of the neuron that it's interacting with. And in reality, the picture is even much more complex than that. There are so many neuroactive components and compounds that can influence how neurotransmitters and neurons behave. So that brings us to the end of our lecture for this week. I hope you enjoyed the exploration of the cortices, the organization of the cortices, the gross anatomy of the brain, as well as the role and function of some of the key neurotransmitters for understanding our brain and the behavior that results from our brain function. You take care for this week, and I will see you next week. You take care. Bye-bye.